Pleasure to be here with you today to tell you a little bit about the work that I've been doing in the Clock Lab at Yale. Motion perception is crucial for survival. It is crucial for a lioness hunting its prey, who's trying to elegantly get away. It is crucial for us trying to cross a busy street where you have multiple vehicles coming at us at different speeds and from varying distances. So given how crucial motion perception is, it's perhaps a little bit surprising that the brain circuitry that we have to do this motion computations sometimes makes mistakes. And we see motion in patterns like this one, where it is in fact no motion at all. And I invite you now to experience that illusion of motion. Let's see if this works. This is a very large auditorium, so doing psychophysics in such conditions is, is bound for disaster. But let's try. Just look in my direction, and I'll flash this pattern in hopefully the visual periphery of some of you. Here we go. Look in my direction. What I hope you should be seeing while the pattern is on the screen is slow clockwise rotation of the pattern. Maybe some of you saw it. It's hard, but it is in fact true. So this animation, and I'll show you data. This animation actually contains another illusion. This one is easier to see if you look straight at a pattern this time around. And you should see anti-clockwise rotation of the pattern as the pattern disappears. And this is quite a bit stronger. Something that is interesting about illusory motion from stationary patterns is that other animals also see it. For example, this cat, it's looking at a pattern that is related to the one I just showed you. And it certainly seems like it's seeing something is moving and it's trying to catch it. And this is anecdotal, but there is indeed evidence that other vertebrates also see motion in patterns that are stationary. We want to understand the mechanisms for this. So we want to ask, first thing, do fruit flies also see motion in stationary patterns? If they do, first we have a lovely way of measuring behavior. And secondly, with vast array of genetic tools that exist to manipulate and measure specific cell types in the brain of the fly, then we really have a good chance at understanding this illusion. So how do we know if the flies see the illusion? Well, we'll look at their behavior while we show them a modified version of the stimulus. What in particular about their stimulus? Well, there's something that is known for a very long time in the field. If you rotate a pattern around the fly, version of the pattern that they prefer, a stationary version of the moving stimulus that they prefer. And the same thing happens for the other pair. So how do we put these things together? So we have a behavior, we have the responses from these neurons. How can we understand the behavior from the responses of these neurons? Well, one simple model tells us that the direction in which flies turn is dictated by the sign of the subtraction of the activity from B neurons from the activity of A neurons. If we apply this to our stationary pattern, these are the two neurons that are most involved in responding to this pattern. And we know from our behavioral measurements that this behavior is positive, so flies turn towards the right, which suggests that the T4A neurons contribute more to this behavior than T5B neurons, or more generally, that T4 neurons' contribution to behavior outweighs that of T5 neurons. So T4, to remind you, are the on-motion detectors, and T5 are the off-motion detectors. And you'll also notice that T4A and T5B are opposite in two ways. One of them responds preferentially to motion to the right, the other one to motion to the left. One is an on-motion detector, the other is an off-motion detector. And what this model also allows us to do is to plan how we can manipulate this illusion. It suggests that it should be possible to manipulate the illusion. For example, if we silence T5, now T4A is the neuron solely responsible for controlling this behavior, so fly should now turn in the same direction as before, but stronger. And if we silence T4 neurons, now T4B will be the only neuron controlling this behavior, and we should see behavior in the opposite negative direction. And that's precisely what we found. If we silence T4 neurons, now flies turn in the same direction as before, in the same direction as controls, but slightly stronger. And if we silence the T4 neurons, now flies start seeing motion in a stationary pattern in the opposite direction than what they used to. So this is pretty awesome. We've really nailed down a causal mechanism for this illusion in flies. So to explain this illusion, we need three things. We need direction-selective neurons that respond to stationary patterns. We need opponent subtraction of left and right signals. And we need different weightings for light and dark moving edges. None of these is particularly fly-specific, so we thought maybe it's not completely nuts to hypothesize that this could also be the basis for the illusion in humans. So we decided to test that. We asked folks to look at this pattern to tell us, when it's stationary, do you see motion clockwise or counterclockwise? Then we added physical motion to the pattern in one direction or the other and at different speeds until we found the velocity of the pattern that cancels out the illusion of motion in each person. And that's called the illusory velocity, that velocity. This is what we found. This is the range of illusory velocities we found for our 11 participants. And I'll remind you that in flies, the reason why we concluded that on and off asymmetric weighting onto behavior must be involved in this illusion is that when we silenced the on pathway or silenced the off pathway, we were able to manipulate the strength and the direction of the illusion. Can we do something similar with flies? Well, we can't silence neurons in flies, but we can show visual adapters, right? We can do light edge adaptation to decrease the gain of responses in the brain of humans to light edges, or show dark edges in both directions, so there's no, some after effects, to decrease the gain of light edge responsive neurons in the human brain. And so that's what happened. This is what happened when we show light edges to people. We see that the illusory velocity goes down, and in fact, now more than half of the participants see the illusion in the opposite direction, just like we saw in flies. But it will come as no surprise for anyone in this audience that the brain of the human is more complicated than the brain of the fly. And so when we use the dark edge adaptation, contrary to what our model predicted, the illusory velocity also went down. But what I would like you to pay attention is that despite not being fully predicted by our model, the consequences of light edge and dark edge adaptation to the illusion were different, and also that these responses are consistent with a model in humans where light and dark edges could be at the cause of this illusion. In summary, I've shown you that flies see motion in stationary patterns like we do, like other vertebrates do. We found a causal mechanism for this illusion, and it involves motion detectors. We were able to enhance or invert the fly's perception of motion, and we did this in humans as well. And using human psychophysics supports a similar model. With that, I would like to acknowledge the Clark Lab at Yale, in particular my advisor, Damon Clark, my co-authors on this study, Ryosuke and Emilio, and I invite you to check out Ryosuke's poster on Friday. Thank you very much.
So we have time for questions. Also, could the third speaker come up? We need your signature.
Hi, I have two questions. First one is you mentioned that the weights for T4 neurons should be bigger than for T5. Is that consistent with like previous studies? Because I feel like the off signal is like more important for flies.
The off signal is more important? How do you mean?
Like the L2 directed information is generally more than the L1 directed on information.
So I don't mean necessarily that the responses of T4 are larger than responses of T5. I'm saying that whatever happens downstream, the weighting on downstream neurons for T4 and T5 is different. And that's what come out of our results. Our results can only be explained if T4 contributes more to behavior than T5.
Okay. Another question I have is, when you measure the neuron response, it seems like it's a transient response.
It's a transient response.
Yeah.
Oh, well, the x-axis was not time, was space.
Okay. So when you present the stimuli to fly, and they can actually turn for as long as 5 seconds?
That's correct.
Yeah, but...
Well, so if you look at each individual fly, they don't consistently, this is on average a lot, maybe 20 flies. So, but there's certainly a bias for each fly to turn to the right. They wiggle a bit, yeah.
So you mean the neural activity versus the behavior, they are like on for similar temperament?
Yeah, there's a match, yeah, especially for T5 neurons we found. They are pretty sustained. T4 a bit more transient. Yeah, thank you.
Thank you.
I was just wondering about phase invariance in the fly visual processing, like the neurons that you're interrogating here, because I wonder if the difference between human behavior and the fly behavior has something to do with a difference in phase invariance in the two visual systems.
I think that's one key. And
Okay.
we don't know the answer. But
Okay.
what we thought we could do is, take so things that are different between human and flies are a ton. Two of them is that flies have one neuron that responds to on edges, one neuron that responds to off edges. Certainly not the case in humans. Humans have many spatial frequencies, many temporal frequencies. So our guess is that we put together a motion energy model with a bank of filters that we would find. Maybe we'll be able to explain the difference. Mm-hmm.
Do you know why we have the difference between the light edge, dark edge imbalance to begin with?
Whys are difficult, but what we do know, it's first, there are other studies that indicate similar results, but we do know that natural images have an asymmetry between light and dark. So,
So embedded in the natural image statistics?
Exactly.
All right, let's thank Margarida again.
Yep.